So a quick explanation of this lab. We were going to plate out dilutions of the red ADA2- yeast, ADE2-, usually we just pronounce it ADA2-. We were going to plate out those red yeast, and so spread them onto auger plates and then directly expose them to different amounts of UV light. And by amounts, I mean different lengths of time. So we were going to expose one plate for 5 seconds of UV, one plate for 10 seconds, 15 seconds. So increasing dosage of UV in this case means increasing amounts of exposure. We were also going to have plates that had no exposure, so those would be the controls. And this picture shows you, after we incubate the plates, typically what we would see. We would see some red cells, usually quite a few red cells or just a few, and then some white. This plate has quite a few white, but it is the photo that I could find on Google. And so you get an idea that we're going to have a lot of the cells are going to be killed, straight up killed by the UV. Just too much DNA damage, too much for the cell to survive. And so I'm going to give you some data simulating how many red cells and how many white cells we would have gotten more or less from previous semesters' experience. I'm going to give you some data, and you can see how the patterns, how the trends look in terms of survival of cells overall and in terms of numbers of white cells as we expose the plates to increasing amounts of UV. So that is what you're going to do in the data analysis part. So I'll give you some sample data and you can analyze that data and make some conclusions.